Very good. So I think, let's begin. I think this is where I stopped yesterday, right? So just to recap very quickly, we're talking about membrane adsorbers. They are macroporous structures that are used. That are used instead of resin-based particles, right? And the idea is that, if you remember, the main reason for doing this is that in the case of resin particles, you have this poor diffusional resistance, and consequently, you have a dynamic binding capacity that is a function of flow rate, and you have inefficient usage of the resin bed, right? And so we have these membranes where you attach ligands on the pores and you pump convectively through the membrane pores. With the resin, convection is outside, but inside the pores it's by diffusion. That's a big difference, right? So we talked about that. We talked about the fact that typically membranes have lower capacity than resins, because if you look at the surface area available, it's less for membranes. And that is a drawback. And so a niche application is to run in flow-through mode and use it to adsorb or bind contaminants. And contaminants are things like host cell protein, DNA, and virus particles, okay? So I think that's where we got up to. And then we talked about bind and elute. We talked about some of these things. And then we got to this point where, yes, we're talking about single-use membrane adsorbers. And I pointed out that actually membrane adsorbers today are used in flow-through mode, and they're all single-use, okay? So everything is single-use, and it's attractive for small-scale bioprocessing. It also eliminates column packing, which is a major issue, especially got big columns. You've got to pack them in a reproducible way. Eliminate the need for cleaning and so forth, minimizes bioburden. And then this is an example of an interesting membrane. It's made by this company Natrix, which was bought by Millipore, and they use a membrane chromatographic media because they're functionalized polyacrylamide gel supported by inert matrix. So there are many, many new developments in these areas which are looking at trying to increase capacity, because capacity is a limitation for membranes, okay? All right, so let's move on from there then and finish this lecture. Now, in order to enhance membrane performance of membrane adsorber technology, something called laterally flown membrane chromatography device has been devised. By the way, I could maybe when you get these slides, I can add the references, because there are references to this, okay? So this is somebody in McMaster University in Canada who's been working on this. So here's the problem. If you have a membrane device, we talked about the fact that you have fast convective flow, so you're going to have more efficient transport of the species you want to bind to the ligand, and therefore you have higher dynamic binding capacities that are independent of flow rate and so forth. But there's a problem, and that is, and this is true for columns as well, you want to have the same residence time whatever flow path you take through the device, okay? You can't have some fluid going through quickly and other going through slowly, because the one that goes through quickly, that part of the resin or the membrane will reach saturation first, and you'll have breakthrough. And so this, we were talking about pleated membranes and these sort of things, if you were to look at that in detail, you'll find that actually there's a risk that you don't have the same residence time throughout the device. There are ways to bypass. And so this is an idea that someone in Canada developed called laterally fed chromatography. It's a very clever idea. So see, the feed is coming in from here, and so the feed comes from here. This is the membrane. So instead of having the feed coming from the center like this, straight into the membrane, they're going to feed it from the corner, okay? If you have it coming from the center, then it's going to suddenly go out like this, and the flow through here will be faster than what goes through the other parts, and the resistance is not the same. But by doing it this way, either it can go all this way and through there, or it can go straight through and there, and if the resistance for flow through these two channels is about the same, which is the case, because it's empty, right? You actually get a much more uniform distribution, okay? So this person in Canada, who is actually very well known, did this work, and I can give you the reference. So this lateral fed membrane chromatography is a uniform flow distribution across a stack of membranes. And this is still, if you like, so when you're making new devices, typically do lab work, then you've got to do small scale work, then you've got to do validation studies, and this is probably at the stage, thank you, of trying to increase the scale. That's where it is. So it's not complete yet, but I think it's very likely that this will take off because this is a very nice idea. Okay, so that's one example of a new development in this area. Okay, sure. So let's pause at that while they restart the computer. In that case, while they're doing that, can I talk about the homework? No, let me talk about the homework problem.
Yes.
They have.
I have sent that.
Okay, so you will get it soon. So there are 3 problems this time, and what I think we can do is one of them I will explain them now, and we can look at them later while they're waiting for this to start. So there are 3 problems. One we've done already, the uniform red blood, the blood cell problem, right? So there's one problem here that says compare hollow fiber blood oxygenated to the human lung. What are the differences? So that's something we'll start talking about maybe today, if not tomorrow, okay? So that's one thing. And then there's a problem here on a hemodialyzer. So you had a lecture, I think, on dialysis. And there's one other problem that talks about concentration of a protein A buffer solution and so forth, which you should be able to do. So those are the 3 problems. So you can work on them tonight, okay? Let me see what else I do. So we'll finish this lecture, and then I'm wondering, we'll talk about emerging membrane technology. Well, let me talk a little bit about these questions then. So you talked about dialysis anyway, right? Okay, so there's a hemodialyzer problem here, which I think maybe for anyone who's a chemical engineer should be easy enough to solve, because it's looking at resistance in series just like a plate heat exchanger, actually. So if you just replace the heat transfer coefficient with the mass transfer coefficient, you should be able to do it. So maybe you can try that one. For those of you who are not chemical engineers, that's fine. We can discuss it, okay? I think you can certainly, you can talk about hollow fiber blood oxygenators, because we'll cover that. And then the other question is concentration of a protein A in buffer B. You wish to double the concentration, and so it's a typical microfiltration type problem, okay? Yes? Okay. So one says compare hollow fiber blood oxygenated to the human lung. What are the differences? Define the desired properties of an optimized blood oxygenator. I think I'm going to talk a little bit about that today before we finish, so that way you can do that question, okay? Otherwise we're delaying. The next one is the hemodialyzer problem. As I said, if you're a chemical engineering student, you should try it, because it is actually very similar to what you would do in an undergraduate heat transfer problem, but it's a mass transfer problem. So it depends if you can see that, but we can talk about it anyway. And then the last one is this one, that the concentration of a protein A in buffer B is 25 milligram per liter. I think you can read that and do that. That's very similar to the one that we did for blood, but this time it's a protein, okay? So 2 problems you can do overnight, and we're supposed to mark them tomorrow, right? So we will do that. The other one, have a look at. If you can do it, great. If not, we'll go through it in class. At the bottom, determine the outlet concentration of urea in the dialyzer. It got cut off, did it? Okay, sorry. It's at the very bottom. This one has got, it got cut off, yes, it's missing there, sorry. Are we right? Okay, good. So if you could send that, then I'll keep going with this in the meantime. Okay, very good. So let's move on. So commercial anion exchange membranes with quaternary amine functionality are available from Sartorius, from Paul Corporation, from Edinburgh Sigma, and from Cytiva. Okay, so these are commercial anion exchange membranes. Membranes typically have DNA binding capacities greater than 20 milligrams per ml and can provide significant endotoxin removal. And this NatriFlow, that's what I was trying to show you earlier, HDQ, I showed you the SEM of that, provides 5-log removal of both parvovirus, minute virus of mice, and murine leukemia virus from a monoclonal antibody product. Very attractive as part of the overall virus clearance strategy in the purification of mammalian cell-derived products. Okay, so membrane adsorbers are in fact rated for removing these contaminants, and that's the most. And they are one of the polishing steps. Okay, now several companies have developed hybrid or synthetic hybrid filters containing features of depth filters and membrane adsorbers. So now you should be able to put these together, because I think on Tuesday, was it Tuesday, Monday or Tuesday, we talked a lot about depth filters, right? And we talked about how these depth filters actually use size exclusion, they use ion exchange, they use hydrophobic interaction, right, to remove cell debris, but maybe you can also use them to validate virus clearance, and that's what the 3M phase is trying to do. So in fact, membrane adsorbers could be using multimodal ligands, right? But the aim is different. Here you're trying to do a polishing step. So you're removing host cell protein, DNA, virus particles. There's no cell debris. But again, you can see that many of the same features are interlinked here. I also talked about the fact that in virus filtration, you have a pre-filter, and that also could have various mechanisms for removal of product-related contaminants this time, okay? So misfolded protein, aggregates, so forth. And they also use hydrophobic interaction, size exclusion, but mainly hydrophobic interaction and ion exchange, okay? The M-phase AEX and ST-AEX, anion exchange materials developed by 3M, as well as virus pre-filters. For example, ViruSoft Pro Shield is a cation exchange chemistry. Shield H has mixed mode chemistry. Mixed mode means ion exchange and hydrophobic interaction from Millipore Sigma, etc. Oh, Sing and L is a reference. I will give you all the references later, okay? M-phase ST-AEX multi-layer filter containing a non-woven polypropylene with quaternary amine functionality followed by a guanidine functionalized polyamide membrane could replace traditional depth filter and flow through an ion exchange step in the post-protein A purification of several MABs and fusion proteins. So if you could do that, if you could replace, right, the traditional depth filter and an anion exchange step, you've put 2 unit operations into one, okay? So that is the whole future direction of these either membrane adsorbers, depth filters, virus pre-filters, to make them more complex with multiple ways of interacting to remove not just one species but multiple contaminant species. There is, however, you have to be very careful, because in the process you might start removing your biopharmaceutical, your product as well, and that you don't want, right? ViruSoft Pro Shield showed more than 20-fold increase in capacity during virus filtration for heavily fouling MABs if you use this as a pre-filter. Okay, a number of multi-channel chromatography processes have been developed for continuous bioprocessing. So, when we talk about continuous bioprocessing, chromatography is a batch process, right? You have a column, you load it, you wash, you elute, you regenerate, you do the whole thing again. Now, probably you've heard about the fact that people have tried to make this continuous, and so you can use moving beds. That was the first idea. So you have your resin particles, they are moving in one direction, your feed is moving in the other direction, right? And so you have a counter-current flow, and so you have a loading stage, you keep going, you get to a washing stage, elution stage, and then it comes regeneration stage and back. The downside with doing that is that you have to pump resin particles. You have to pump a slurry around the place, and that is difficult to pump. Pumping slurries is difficult to start with because it can get stuck and it can plug, but also you tend to damage the particles, and so the lifetime of these particles is limited. So then people thought about using something called simulated moving bed, right? Which basically means you have 4 columns or 5 columns, whatever you want, and by switching valves, you switch the columns. You don't actually move anything. So while this column is regenerating, the one next to it you're eluting, the one next to it you're washing, the one next to that you're loading, and so forth. And you just switch valves and you can keep this going. So in fact, you've had something loading at all times, okay? So that's another way people tried to come up with this idea of using multi-column chromatography processes for continuous bioprocessing. These systems work cyclically with large variations in product concentration and buffer concentration as the column moves through binding, washing, and elution steps. So the problem with this is, you could say it's continuous, but one thing in the biotech industry, we want to keep things constant. And so when you're eluting, you start to get initially a high product concentration, then it tails off as you get close to the end of the elution step. Then when you start the next one, the same thing happens again. So it's not uniform. That is a downside, okay? A number of column chromatography becomes less attractive as the product titer increases, since the volume of the expensive resin must be increased to accommodate greater product mass. That makes sense, right? Number of efforts to develop continuous capture step that can be connected directly to the outflow from a perfusion bioreactor as part of a truly integrated continuous biomanufacturing process. So what we really like to do is you have your bioreactor, you have a perfusion bioreactor connected to one of those, say, alternating tangential flow systems we talked about. So you're continually taking your product out, okay? And you've got the product, now you'd like to continually load it onto your chromatography column. You don't want to have any sort of holding tank. You'd like to have it going straight. And so with doing simulated moving bed, it's tricky because you have to keep switching valves. And as I told you, the elution peaks, you get a peak and then it comes down again. And so you may need to have a holding tank every so often. So one idea, a very recent idea, is this idea of membrane technology can play a critical role in this application with actual separation purification of the protein product accompanied by selective precipitation or continuous countercurrent tangential chromatography. So those are two membrane processes, if you like, membrane enhanced processes for continuous. Let's talk about those briefly. So here's continuous countercurrent tangential chromatography. And so the idea is this, and I just wanted to introduce it briefly, because it's likely that in the future there will be many more of these kind of combined processes where you have traditional chromatography steps enhanced by a membrane. So if you look at this, continuous countercurrent tangential chromatography uses ion exchange, affinity, or mixed mode resin particles. The particles flow in a slurry with the solid liquid separations accompanied using a hollow fiber. When used in bind and elute mode, product binds to the resin with soluble impurities, host cell DNA removed through the membrane during washing. So let's see what happens here. If you look at this, in this particular example, you can see that you have a static mixer here, okay? You have your solution coming here. This is actually, okay, this is the wrong, hang on, let me do one thing. This is actually okay, this is actually refers to the next one I'm going to talk about. So let me come to that figure in a second. Just a second, let me come to that in a second. Okay, so the product collected in the permeate by adding an appropriate elution buffer to reverse binding interactions. Each step in the chromatography process, you've got binding, washing, elution, stripping, and regeneration. Use a combination of a hollow fiber membrane module and a static mixer, with the mixer providing the required residence time for binding to the protein, to the resin. The hollow fiber membrane module is arranged in countercurrent configuration, analogous to that shown in the figure. So now let me go back to the figure. Why will it move? What happened? No? Why can't I make it move? So this screen has frozen now somehow. Why is that? That one is not showing, right? The right one? What happened? That one's not moving. Do I have to do this? This one's wrong here. No? I don't know. Okay, now it's right. Okay, thank you. Okay, so now I can explain this. So let's look at this. So you have basically your feed coming in here. You have static mixing stage. You can run it through a hollow fiber system. You use the resin particles to remove. You can have a washing step and so forth, okay? So basically you're using the hollow fibers to to selectively remove your contaminants as you're continually running through this step. Okay. Okay, so there are a number of advantages, distinct advantages compared to traditional column chromatography. Operated continuously in a truly steady state, ideal for incorporation in integrated continuous bioprocess. Since the resin is used as a flowing slurry, the pressure drop for this continuous countercurrent tangential chromatography is less than 15 psi, enabling the flow path to be made completely disposable plastic materials in contrast to stainless steel columns used in commercial chromatography. So if you can use plastic disposable material, that's much cheaper, and people want to use single-use devices. So again, here's a possibility. This sort of countercurrent tangential chromatography can be used in the development of single-use biomanufacturing facilities, enhancing flexibility and reducing capital costs. It has been shown to give productivities as high as 100 gram of MAB per liter of resin per hour. That's actually very high capacity, okay? High productivity is a result of the resin being fully utilized at all times. So that's a very important point. Even if you are using simulated bed chromatography or adsorption, the column is here, so you start to load the column, right? And you start to get, say, saturation occurring at the bottom, and it moves up to the top. But you will never get all the resin fully loaded, because as soon as you start to get outlet concentration, say 1%, 2%, 5%, of the feed concentration, you must stop, right? Otherwise, you're losing too much protein. But it means that the top is not at equilibrium. It has not been fully saturated. So you are not completely using the resin. And that is always a problem. These resins are expensive, and anyway, you want to get maximum capacity. So that is the advantage if you have this flowing through in a hollow fiber. Now, the downside I told you a moment ago is that pumping a slurry is always difficult, right? The idea here is it's flowing inside a hollow fiber, and so it's less likely to be damaged or to be broken due to shear stresses and things, okay? But that is a trade-off. Okay. So here's another example of this. So here are the resin particles, and basically continuous countercurrent tangential chromatography for impurity removal. Filled ovals form a bound monoclonal antibody, and you can see these are in the Y shape, and then the contaminants are being taken out through the membrane, okay? So this kind of combined hollow fibers or membranes and resin particles is likely to be a direction in the future, okay? Oh, one other reason why this is an interesting way to go, all of this is FDA or regulatory authority approved, right? Resin particles are approved. Hollow fibers are approved. So you don't have such a big activation energy, if you like, to get all of this approved again, you see? So that's another reason why people are looking at these sort of combined unit operations. Okay, so I think we have basically finished everything now, right? We talked about depth filtration, we talked about sterile filtration, we talked about membrane adsorbers that fit in here, we talked about virus filtration, we talked about ultrafiltration, diafiltration. That is not to say that there aren't non-membrane processes, which we didn't talk about. So I told you typically the capture step is going to be affinity A, and it's a resin step. Typically, there are two polishing steps at least, and only one of them is usually a membrane adsorber. The other is going to be a packed bed, okay? And then there are other things, low pH hold steps and so forth. So that is not to say that the whole process is run just with a membrane, okay? But those are the membrane steps, which is what this course is about. Okay, so future directions, I talked about some of these. Single-use devices. Continuous biomanufacturing means continuous operation. We have to come up with devices that can be used in continuous operation mode. Bind and elute. So I told you, at the moment the capture step is protein A is resin. And there are attempts to try and make that a membrane process, okay? But bind and elute chromatography is something that is not done very much with membranes yet. And then membrane enhanced separations, and I just talked about some of those, okay? So, any questions on that? That is the end of that lecture, which was, yes? Yes? Oh, sure. The lateral column, 1 second. It'll come in under pressure, yes, yes.
First, when there is a high pressure there,
Yeah.
won't the fluid directly come from there?
I agree. It'll go through here, but then from here it has to go through here to come out. Okay, but the pressure drop, what you want to do is have the pressure drop for that path to be the same as any one of these other paths. So the idea is that if the pressure drop across the membrane is the same, wherever it is, then these are always going to be the same because the lateral distance it has to go is the same, whether it's from the top or from the bottom.
What if it shouldn't slide, it's slanted?
Yeah, then it shouldn't slant. Yes, I agree with you. So it has to be designed very carefully. Yes. Now, the reality is if it's slightly slanted, it really depends on what is the pressure, the relative pressure drop through here to this. If it's a small difference, it doesn't matter. But if it's large compared to the overall pressure drop, it's a problem. But yes, yes. But this is still much easier than packing a column. Okay, but yeah, you're right. Any other questions? Yes. So if it's for removal of contaminants like endotoxins, virus particles, it actually is much more efficient. If you can get the how can I put it? If you can get the flow and things worked out correctly, it's much more efficient because you don't have pore diffusional resistance. So it's much more efficient. But for bind and elute, it's not. Okay. If you want to bind your product and elute it, it's not. Okay, and we didn't get into that, but one of the reasons is the capacity limitation I told you. See, there's a lot of product, that's a problem. There's another reason too. It turns out, this is a very interesting point, that actually if you take the same ligand and attach it to a resin and then to a membrane, the idea is you bind but that you can easily elute, right? Sometimes it's very difficult to elute from a membrane. It's stuck there, even though it's the same ligand. And the thought is that these are due to geometrical effects. So in the membrane, you might have multiple ligands attaching to the same species, same product, solute, whatever, and that makes it more difficult to release. But this has been observed. That's another problem. Okay. Okay. All right. So let me then stop sharing this one, and we'll move on. Sorry, where's my glass? All right, so then today you had a lecture on, I think, hemodialysis. Good. And so now let me move on to my next one. So remember, we changed order a bit. We did virus filtration already. And so then I need to move to emerging technologies, which is this one. So let me share this. Okay. Good. So in this, what I'd like to do in this lecture is talk a little bit about some of these emerging areas. So basically, we should have covered all of this stuff, and we're now working on these two, okay? All right, so here is what I'd like to talk about. Something called high-performance tangential flow filtration, HPTFF. Single-use tangential flow filtration. We've talked about single-use chromatography devices. I'll talk a little bit about single-use tangential flow filtration. Under continuous bioprocessing, I'd like to talk a little bit about alternating tangential flow filtration. We've referred to it a bit, but I'll talk a little bit about that. Continuous precipitation, that's a membrane-enabled process or a membrane combined process. Continuous countercurrent tangential flow chromatography I talked about already. A single-pass tangential flow filtration, and continuous diafiltration. So those are some of the, if you like, emerging technologies, but again, they're all based on existing technology, and that's important because otherwise it'll take a very long time to get approval for them. Okay? So let's move on to the first one. So this is our whole setup again, but we just went through that, so let me skip that. Okay, so high-performance tangential flow filtration. What is this? And this is a very example because it seems like a fairly nice technology. High-performance tangential flow filtration is an emerging technology that uses semipermeable membranes for separation of proteins without limit to their size. This contrasts with conventional ultrafiltration processes that require a 10-fold difference in effective size for separation. I think when we were talking about this earlier, one of you mentioned, I asked, what do you use ultrafiltration for? Somebody talked about proteins, separating proteins, and I said, no, not at the moment. It's only actually buffer exchange and concentration, which is true. However, and it's true for this reason, because using conventional ultrafiltration, you need at least 1 order of magnitude difference in molecular weight to get any sort of decent fractionation, where you reject one species that the other goes through. If it's less than that, you will get imperfect separation, okay? So too much of the one species will get into the permeate and so forth. High performance tangential flow filtration tries to overcome this problem. So you can now fractionate that may be a much, much closer in molecular weight. That is the aim, okay? So it has been used to separate monomers from oligomers, so they are very close in molecular weight, based on differences in size, protein variants differing at only a single amino acid residue, antigen binding fragment from a similar size impurity, and then we have done work where we looked at removing small viruses from DNA host cell proteins. Again, very close in size, okay? HPTFF can be used throughout the purification process to remove specific impurities, protein, DNA, or any toxins, and or eliminate protein, oligomers, and degradation products. It can conduct simultaneous purification, concentration, and buffer exchange. So you can combine all of these into one, and we often talk about process intensification. Very important. If you can do that, you save a lot of money, okay? So here is the idea. Oh, sorry. Oh, it got stuck again. Why is that? Why is it stuck again? I don't know why it's stuck again, right? Okay, now it's working. That's fine. It just seems to be slow today. Okay. Never mind. Okay. All right, let's keep going. Okay, so here is the idea. So here is a conventional operation, okay? And I talked about this earlier. In conventional operation, you have your feed going this way. You're pulling your permeate or filtrate through the membrane, and retentate goes through. And as you flow along here, there will be a pressure drop on the retentate side and the filtrate side. Do you agree? As you flow through. The transmembrane pressure then, we typically say, is the average of this one and this one minus this, and we often assume that this is just constant, right? And we say minus. But that's not true. Actually, the transmembrane pressure drop is this difference here at this point. It's this difference here at that point. So there is a change in transmembrane pressure drop as you go through this device. And a change in transmembrane pressure drop means what? A change in the amount of concentration polarization, a change in the amount of fouling, and the change in the resistance to flow, because we have got a different convective force towards that. What we'd like to do is have them all exactly the same. So across the module, the TM is always this difference, and it changes. If you can have a constant transmembrane pressure drop across the length of the module, you will get a far more efficient separation, because now you're not varying the conditions along the module, right? So you'll be able to get a much tighter, a much more clean separation, and you'll be able to separate species that are much closer in size. That is the whole idea. So how can we do that is the question. How can you get this, okay? And the idea is this, as they show you here. You take permeate, and some of the permeate you pump back along here in a co-flow mode, okay? So you have co-flow, not countercurrent flow, co-flow of the permeate and the feed. Therefore, you're trying to match these two pressure drops. That's what you're trying to do, okay? So by having flow, you keep the pressure drop along the length of the channel the same. So there's a price you pay, because actually the amount of filtrate you produce per pass now, right? Because some of it's being recycled. But that is the idea of high-performance tangential filtration. And there are patents on this, and there are a number of papers on it, okay? So now I guess this will move slowly. So high-performance tangential flow filtration provides high selectivity by operating in what's called a pressure-dependent regime at or below the transition point. Plot of filter flux versus transmembrane pressure. So if you remember, we talked about this, and we said that if you look at the plot of say a flux versus pressure, it does something like this, and here you get to the pressure independent region. You want to operate in this range, okay, in the pressure region. Low current flow, selectivity is a function of local filtrate flux and local transmembrane pressure. Selectivity can be further improved by maintaining nearly uniform transmembrane pressure throughout the module. Conventional membrane modules have large variation in transmembrane pressure drop due to parasitic pressure losses. That's the pressure loss as you go through with flow along the retentate channel. Okay. Co-current flow of filtrate using a recirculation pump generates a pressure gradient in filtrate channel that balances the gradient in the retentate channel. That's what you're trying to do. Okay. So look at one other example here for this one. So this is an example, and again I can give you all the references and do that tomorrow. So here you're looking at two different plots. They are the open symbols and the symbols. So the diamonds here, the diamonds are given here. So the open symbols refer to this, and the filled symbols refer here. Okay. And what you're trying to show is that the selectivity, which is defined as the ratio of sieving coefficient for the impurity to the product, is therefore larger using co-current flow. Okay? So the antibody solution with Chinese hamster ovary impurities without co-current flow is the symbol. So this is without co-current flow, and this is with. Okay? And so if the ratio of what's in the filtrate to what's in the feed, here it's almost 1. So that means there was no separation. But here, significant separation. So you can see the tremendous benefits you get from this, this high performance tangential flow. Potentially, it's very nice. Now, you are paying a price because you do have an extra pump, right? So it's complicated. You have to control that pump, and you also produce your product at a slightly slower rate because some of your permeate is being recycled. Okay. A few other things about high performance tangential flow filtration. Buffer effects. Significant improvements in performance can be obtained by controlling the buffer pH. That's better. Increase the performance by controlling buffer pH ionic strength to maximize differences in effective volume and impurities. So remember, what we're trying to do is get very tight situations now, species that are very close to each other by maintaining this very constant pressure drop. But I told you that the size of a protein depends on conditions, right? Protein is a hydrodynamic radiation, may have other charged species associated with it. So by controlling buffer conditions, you can actually try to maximize the difference in size between your contaminant species and what you want. Buffer effects are very important in this particular separation. As discussed yesterday, the effective volume of a charged protein accounts for the presence of a electrical double layer surrounding the protein, increasing charge, reducing solution ionic strength, volume, thus reducing protein membrane. So you can play with all of these to optimize your separation, and then you can do fractionation, oligomers and dimers from monomers and so forth. Optical performance is typically attained by operating close to the isoelectric point of the lower molecular weight protein, a relatively low solution, to minimize electrostatic exclusion of the more rich species. So the smaller molecular species is what's going to go through the membrane. If you work at the isoelectric point of that, there should be very double layer around it, right? Because there's no charge interaction. So you minimize the size of that. The other one will have some double layer. You're trying to maximize the size difference between them. Okay. Membrane charge. Again, charge affects membrane that has an electric retention or passage of species. If the species have the same charge, you're more likely to reject them. Charged membrane provide much retention of positively charged protein than will a negatively charged one or neutral membrane. I think that's fairly straightforward, right? Membrane pore size distribution is important. Membrane pore size distribution affects selectivity, solute sieving coefficients, rate distribution. And so elimination of large defects and reduction of the breadth distribution significantly improve performance. Okay. A variety of membranes have been used and geometries used. Those membranes that have been surface covalently attach either quaternary, amine, or sulfonic acid groups tend to be the desired ones. Regenerate cellulose to use non-specific adsorption, and you can modify it very easily. The downside is it's not so varying pH conditions, so you be careful. Membrane cassettes for HTP-FF are similar to ultrafiltration modules, specially designed to linear scaling from small modules suitable for processing, say less than 50 centimeters squared, commercial devices capable of 15,000 liters in 3 hours. Okay, so that's quite a range. Okay, so equipment. TFF uses same filtration, co-current filtration accompanied by addition of a co-flow loop and filtrate side. Here's an process, so it doesn't have to be one stage, it can be here's an example of a two stage. To see what happens, this is recycle tank 1, your tank from feed tank 1, this is 1, it goes into the membrane device and your retentate comes back. Okay, the permeate comes out of here, okay, and this goes to recycle tank 2, okay. The retentate from that comes back in here, and then the permeate from here in what's happening. So, so basically you can link these two, and what you end up doing is concentrating things in here and concentrating things here, okay. Like this, you cascade sometime and look at the questions, but you can develop cascades like this to fractionate many different proteins of different okay, let's see. I think that's the end for HP tangential flow filtration. So if you think about it, we've talked about single-use depth filters, single-use virus filters, single-use sterile filters, single-use membrane adsorbers, all of which are not using any recycle. There's no recycle of the feed. There's no flow or counter. There's none of this. But if you want to do single-use tangential filtration, this is different, right? Because this is one where you're recycling the retentate. So UFDF used concentration and buffer exchange, we saw that earlier, in formulating all bioreactors. They employ membranes with pore 5 nanometers, provide retention of the product and relative of lower molecular weight impurities. Systems, large, we saw these photos earlier, large stainless steel membrane fed from large stainless steel tanks, right? And that's all expensive. Single-use systems are typically fed from disposable plastic bags, okay, with volume. That's pretty for plastic, and have fully disposable flow paths made of a tubing and sterile connector. So basically, you can take this whole plastic tubing set that's totally sterilized. It's closed, hang it on the equipment, and, okay. So it's very easy. Systems typically for pressure flow, temperature, and conductivity, and UV absorbance. So it's all use and throw away. That's the idea. So UFDF can be used to remove a wide range of extract leachables might be generated use bags and other systems used throughout the biomanufacturing process. So this is important because plastic instead of stainless steel tank things, you've got to worry about leachables. Okay, I think I mentioned to somebody I was talking to. When you're developing or you need to be concerned and be leaching, and so devices now you've got to make sure leachables are removed. Okay, data with acetonitrile, benzyl alcohol, and these other hexanoic acid, all of these 1,000 reduction in concentration of these model extractables or leachables within 9 dia volumes consistent with standard model during diafiltration. A dia numbers what that is, you need to know it for the anyway. It's a feed volume, one feed volume. Okay, so 9 times the feed volume you would have to use, and actually we've seen this before. Contaminant when you're doing diafiltration, exponential decrease as shown here, where ND is the ratio of feed volume, constant volume is the observed sieving coefficient, equal 1, then just negative ND. Okay. So equal to the ratio of the solid concentration of the permeate and the retentate. Good. 1 for small electrostatic reachables. Okay. So here's an interesting figure. There is so much interest in single volume, in single use devices. So this is a reusable device, and this is the amount of time required. So the blue time here, the gray time is what you need for sanitization. This is for equilibrium. Purple is actually running UFDF, and then the red one is clean, uninstall, and you're going to again. Okay, so that's the time taken. Feed volume for a single, you have about the same time for equilibration, but you don't the run time and the time for. So you can see there's a reduction in time. That's a big advantage of this. Okay, and in the example, that is a very big. So comparison processing time for single usable final form recombinant protein. Single use systems pre-sanitized using gamma irradiation allowed to be used without any initial sanitization step, including to remove chemicals used for sanitization. Post-use cleaning cycle to generate membrane product can simply be and recover. So that's the advantage of single. Tangential flow, specifically for bioseparation applications, have been made. These single-use devices have been made. Here are some examples. Single-use system, Moebius Flex Ready solution for tangential flow, TangeX, so millipore, and so forth. All gamma sterilized and provided in preservative-free water with aseptic connectors. The water is sterile that they're served in. It's aseptic. You can just run it. Okay. Greatly facilitating their use as a plate processing solutions. Single-use hollow, those are all flat sheet hollow modules from Cytiva designed to mimic corresponding reusable systems, comparable feed side pressure drops and mass transfer coefficient, but with aseptic connectors to facilitate their application use systems. By using equivalent flow geometries, length and channel height of 5 years, including space elements. Basically, you're trying to model what happens is a reusable device, but it's in a single-use device. Okay, and these are all available. Single-use device, it's possible to develop ultrafiltration modules specifically targeted to single-use applications. Use polyphone, hollow fiber, hemolysis cartridges operated at low filtration for protein concentration single pass. Modules are sterilized format with aseptic using tubing designed for extracorporeal blood circuit. Let me just try and explain this. Before this, we were talking about single-use device. Single-use device, not necessarily for continuous biomanufacturing. They are single units, but just single use. Now, if we want to go to continuous biomanufacturing, ultrafiltration is really a continuous process because you're recycling all the time. Recycling, you bring in product continuously, actually don't really, that's not truly equilibrium. And if there's any hold up anywhere, you're in trouble. So it's not really such an easy way to do a single-use device. What I prefer to have is come in through your UF module, and then the retentate go off somewhere else, and then the product continue, and you don't have any recycle. That's what you'd like to have. So the suggestion is that this could be done, and I'll show them in the next slide. Now, the hemodialysis cartridge dialysis. So it's a dialysis process, not an ultrafiltration process. So does anyone know the difference? Force. Hemodialysis, it's a concentrating force, right? There's also some filtration that uses both, and we have time, we'll talk about it tomorrow. Okay, so that's what the aim is with to do. Hemodialyzers are sold, sterilized in here. Okay, so current bio. There's a on the next one. I didn't put the diagram. Never mind. Current bioprocessing operations are performed largely in batch mode. Concerns about biomanufacturing and need for for greater manufacturing flexibility, interest in this continuous bioprocessing. Capital and equipment costs are important. Increase in productivity. All equipment is in use all the time. Greater flexibility due to more efficient utilization of quality due to state operation. The quality continuous process shorter and times distributed batch leading to variable. So actually all of these are fair. The advantage by manufacturing cheaper, you don't have to set up, take down, and also importantly, you get uniform product quality. Those are the reasons for trying to do this. And so, as I said here, there is a single, okay. Pressure on biomanufacturers to reduce the price of biopharmaceuticals. That's an important point. There's great pressure on that because I told you some of these drugs are like $1 million a dose, the most ones. Combat the growing overall cost of healthcare and increase the availability of novel therapeutics in developing. Lower cost manufacturing providing increased competition for established pharmaceutical companies, especially making biosimilars. Estimated integrated continuous biomanufacturing processes, this is very important, would 5% lower cost for production of monoclonal antibody products. And that's an estimate. Yes, that's a huge to start working on continuous biomanufacturing. If you can see 55% of the cost. And on top of all the other benefits like uniform product quality, enhanced manufacturing facility designed to produce multiple products, and so forth. Now, continuous cell culture technology, fusion bioreactors developed in 3 decades. This did not talk much about cell culture because that was not the aim, it was bioseparations. But this, you have a reactor, it's called fed batch, it's running a new growth medium every so often, and that's about it. Okay? At some point you stop and you harvest or do clarification, you recover your product. Perfusion reactor is different, you want to continually add growth medium, continuously extract waste products as well as your product. Okay? So continuous cell culture, and this has been done for more than 3 decades using perfusion. Perfusion bioreactors operate with continuous addition of continuous removal of periodic purging of cells, debris to maintain bioreactor volume and cell density. For production of highly labile products that are susceptible to degradation and proteolysis. So what I mean by short residence, product is produced, it doesn't stay in the bioreactor too long, continuously removing it. If it stays there too long, you could be getting other sorts of enzymes and that degrade the product. That is the concern. Perfusion cell density is nearly 10-fold greater than batch bioreactors. That's also important. Higher cell density, higher product titer. Okay, leading to enhanced product. Fusion bioreactor monoclonal antibody production is now well established. However, the necessity to recycle cells back to the perfusion bioreactor places a significant constraints on clarification step as cells for long-term bioreactors. Recycling this, you must put the cells back. Problem must be a depth filtration process. You cannot put cells back. Right? Tangential that, but it is sterile. The next problem is that you don't want the cells to go through kind of a very hard flow path. There's a lot of if you don't not so viable, they may grow so well. You don't want to change the character of cells. This is all making it very difficult. This is a challenge. Irrigation and depth filters just don't work. Okay. Cell recycle and clarification using tangential flow filtration with a large ID in a diameter, hollow fiber membrane. Retentate stream, which contains the retained cells, returned directly to the bioreactor, while the permeate stream, which contains the secreted product, is taken for purification. Okay, and they operate at constant flow. Okay, on development of membrane processes that reduce the extent of fouling and minimize product retention, allowing a single module to be used for multiple weeks without cleaning or replacement. That's quite a challenge. You want to use for multiple weeks. So tangential flow filtration, you're using large diameter fibers, but nevertheless you will be fouling eventually. And if it doesn't last a month, because now you've got to switch these out. That's a possibility of having a sterile operation. If it's non-sterile operation, you'll kill whatever is in the bioreactor, okay? Antibody transmission through microfiltrant membranes is 100% at the start, but significantly product retention over the course of operation as the significant changes. Due to fouling associated with cell debris that is approximately 100 in size, smaller than the 200 nanometer pore size of the membrane. So these things are just small pores, you start to see product retention, and that's no good. Reduce generation of debris by using low shear centrifugation. In contrast to peristaltic pumps used for traditional flow filtration, you can do that. Evidence of membrane fouling micelles formed by antifoam agents used in minimizing reactors. So that's okay. And so one idea, as I said, you use large pore size for micro, very large pore operation, nearly 100% product transmission. The problem then is you have a turbid permeate. There is a possibility of getting into means you cannot use it directly into the capture column, or you'll start plugging up your chromatography resin. So you have to put an additional depth filter or something. So that's complicated. Improve performance using something called alternating tangential flow filtration. Use large in a diameter hollow fiber membrane. Flow is pumped through in an alternating fashion by diaphragm that withdraws, turns the cell, the bioreactor in a cyclical fashion. Cycle times are the order of 1 minute. Cell damage due to high shear, there's no peristaltic pump anymore. Alternating flow direction can unclog fibers that become blocked cell and cell at the inlet. It occurs in both directions. Alternating tangential flow systems have only a single the bioreactor, which helps maintain stability. And I showed, I have it here. Okay, let's first, and then I'll come back. Okay, let me explain this. If you look at this, okay, so here is conventional tangential flow. So you have a this is a peristaltic pump. Okay, you have stuff here. It's pumped here, pumped fibers. You remove your filtrate, and then it goes back to your bioreactor here. The disadvantage is that this starts to foul eventually, so you start product rejection. Peristaltic pump can damage the cells, and you have 2 connections to your bioreactor. And the more connections you have, the more likely you are to have non-sterile conditions if anything goes wrong. So with alternating tangential flow, this is the bioreactor. The diaphragm pump sucks stuff this way, so it pulls liquid through here. As it pulls the liquid through cells as well, the filtrate goes out here. You stop, and then you push air this way, and the diaphragm moves. This is the diaphragm. It moves in the other direction, pushes the liquid back. So it pushes the liquid cells, everything back into the bioreactor. You withdraw permeate, goes through. So it's back and forth, back and forth, back and forth. This reversal in flow helps remove concentration polarization, and therefore you get much fouling. Okay. You also only have one connection to the bioreactor, and you have far less because you're not pumping the cells through a peristaltic pump with constriction, right? So all of these mean that this system is actually very nice and works very well with perfusion bioreactors, and this is called alternating tangential flow. Okay. I think I have to say one more thing here. ATF, So system operated at very low flux, about 0.3 to 2 micrometer per second, 1 to 5 LMH, using feed flux rate normalized by the area to 800 LMH. So remember, I told you in continuous biomanufacturing, speed is not important for a long time, but we can run slowly. That's the advantage. Combination of low flux, high feed flow rates, typical shear rates of 1 to 5 seconds, and high membrane permeability generates something called Starling recirculation flow in the module with the large axial pressure drop flow in the retentate. Let me see if I can explain that in the previous slide. So what it's saying is that if you're pumping this way and you have a large flux going out, okay, at some point you may actually, because remember you need conservation of mass, the volume decreases here, you start to get reverse flow and going back in from the other direction. Okay, Now, the downside of that is that the permeate to recover is less because some of it's going back in. But the good thing, it's actually back flushing the membrane and cleaning the membrane. And as mentioned earlier, it does operate at very low permeate flows, but that's okay because we don't mind. We want to run it as long as possible. So that's that occurs in this. Okay, this flow. Sorry, what here? Okay. Backflushing can clean much of the membrane length since the alternating flow shifts the location of backflushing from one end to the other. Okay? All right. A few more things. We talked about some of this already. So multi-column chromatography processes, this is membrane-enhanced separations, developed for continuous bioprocessing. These simple systems work by cyclical with a large variation in product concentration. Column chromatography becomes less attractive as the product tighter increases, becomes more expensive, and so we are looking at chromatography can play a critical role here. And I've talked to you quite a bit about membrane chromatography and the advantages here. So I'll move on from that one. Continuous precipitation, I think, is the next, right here. Continuous precipitation is an interesting idea. So the idea is this. It is possible, so this is something that is not done much these days, but there's certain lot of interest in this. It's possible to purify a monoclonal antibody products by precipitation from clarified cell culture fluid using a variety of salts and polymers. So you're going to precipitate your antibody, your protein, recover it as a precipitate, then you've got to resolubilize it. Okay, that's the trick. Continuous precipitation reactors can provide more uniform residence time and precipitate morphology. Host cell proteins and DNA can then be removed by washing with a very, very easy, right? You've got to precipitate, so you remove that and you can wash away everything else. With precipitated protein recovered by tangential flow microfiltration, while the soluble impurities are removed in the permeate. So that's a nice system potentially. MABs, monoclonal antibodies, successfully precipitated using a variety of precipitants and flocculants, including, and probably caprylic acid is probably the one that people like to use the most, but calcium chloride, zinc chloride, polymers, polyethylene glycol. You could also use anionic, cationic polymers that are used in water treatment, but I told you the problem with that is they are not regulatory authority approved, not FDA or other approved, and so they don't meet the specifications. You might have some of the monomer there, which is highly toxic, you see. But these things that I mentioned there are approved for use in biopharmaceutical manufacturing, especially caprylic acid. Okay? What's next? For continuous precipitation, high degree of purification achieved using countercurrent stage microfiltration modules, MAB precipitation driven by a combination of cross-linking agent, precipitate approximately 2% solids dewatered by microfiltration. Okay. And so here's another figure that was similar to the previous one I showed, where you can combine these units and run them in a series like that. And you can have multiple steps. You can have one, you can do three, you can do it many stages. Okay. Okay, high degree achieved by countercurrent stages. Here, in extent of impurity removal, you can actually calculate here mathematically if you wanted to, and you can therefore determine the number of stages that are required. Okay, for those of you that have studied things like liquid extraction and so forth, the analysis is very similar, very, very similar. Okay. For those of you that haven't studied that, basically there are mathematical ways of linking up a number of stages and actually coming up with factors. You can here see here, in this case, R is going to be the extent of impurity removal, and it's given by alpha to the n plus 1, where n is the number of stages and so forth, and alpha is going to be a separation factor and so forth. Okay. Okay, ultrafiltration modules, we're still talking about single pass tangential flow filtration. Ultrafiltration modules operate at relatively low conversion ratio to permeate feed inlet flow rate. Required degree of concentration achieved by continually recycling the retentate through the module, right? Each pass, what you take out is relatively little. What you end up doing is recycling a lot, right? Inherently a batch process, its desired target product concentration not obtained until the end of the process. That's important. So, in a continuous process, something runs through into operation and you're continually producing product. In ultrafiltration, that's not true because even though you're continually recycling, the final product is only at the end of the process. Before that, you don't have the right concentration, right? So it's not a continuous process in that sense. Several companies have developed ultrafiltration modules for continuous bioprocessing, desired concentration factor or volume reduction obtained in a single pass. So this is quite interesting. So instead of having to recycle in one pass, you get to the concentration you want. So single pass tangential flow filtration, or SPTFF, used for inline concentration of feed between unit operations. Remember that diagram I showed you, and we have steps in between? So you could use a single pass ultrafiltration and go to the next unit operation. And they are made today, they are available. SPTFF used in final formulation of biotherapeutics and single pass operation enabling higher product recoveries, particularly for high concentration antibody formulations. Absence of recycle stream can prevent aggregation or denaturation, and that's very important. So here's an example of one, and there are various things that people have tried. So here's a single pass process, and this is actually sold and is available, and there are other ways to do it as well. So here, imagine this. So initially, your feed comes into this module, and just for argument's sake, there are 3 channels, but there could be 60 channels, whatever you want, okay? So it does a single pass through this. There's no recycle, single pass, okay? And then it comes around here and goes through again, all right? And this time you have 2 channels. You have a reduction in the number of channels. Why? Because you've removed permeate. So your feed volume has decreased. So if you want to have the same fluxes and the same wall shear rates, you have to reduce the number of channels. So you come through here, and then you can reduce this to 1 channel. This is just for argument's sake, right? And then it comes out there, and then it can come right the way out, okay? So it just goes through the unit once, but the way the unit is designed, you might have, say, this is showing 3, but maybe 10 channels to 5 channels to 2 channels. So it goes like this through the whole unit, and that's it. There is no going back to the feed tank. So it is a truly single pass device. Schematic diagram of flow path in a multi-cassette SPTFF module with sequentially decreasing number of parallel channels, 3 to 2 to 1, okay? That's one design. There are other designs as well. Okay, what about continuous diafiltration then? Okay, and I think I've almost finished with this lecture then. So diafiltration is conducted using constant volume batch system with high level of buffer exchange requiring between 5 to 10 diavolumes, where we know what a diavolume is, right? It's the feed volume. Now, so drawbacks with diafiltration. One is it's not continuous because you're continually going back to the feed tank. And number 2, you're using a lot of buffer. So the question comes up, can I use less buffer? Can I use less diavolumes and get to my required buffer concentration, right? And can I make it continuous and not a batch process? It requires several hours of operation and close to 100 pump passes with diafiltered product not attained until the end of the process again, and pump passes can damage our protein. Alternative approach for continuous buffer exchange is to use a simple inline dilution of the feed followed by continuous SPTFF. However, in 1,000-fold reduction in concentration of particular salt or impurity would require a ratio of 999. What that means is that we could run this system. Let me see if sorry, that's not what I wanted. Go the other way. Yeah, we could use this system. This was a single pass, and we could keep adding throughout this our diluent all the way through. But that's really not very effective, okay? You end up using even more volume than you would if you use constant volume diafiltration than we do now, okay? And so that's what that was telling you. So a series of sequential dilutions and concentration steps can be used, but you still have higher buffer usage. So that is, at the moment, we have 1 feed tank. We are adding buffer or diluent as we are removing permeate. So this is saying, why not take this, add say 3 liters, add 1 diavolume, then run it, and then add another diavolume, and then run it, and then, but you see, if you do it continuously, you have that exponential decrease in concentration. If you put it all at once, you get a step decrease like this. You'll find you use more buffer, okay? The exponential will still be quicker. We'll be using less buffer. So that also doesn't work that well. So here's an idea that actually works very nicely. And so for those of you who are chemical engineers, or you should find this actually very logical. More effective operation achieved by arranging single pass tangential flow filtration modules in countercurrent configuration to increase effectiveness of staging. Remember, countercurrent extraction or countercurrent any of these processes always is more efficient, right? We learned that in undergraduate classes, right? In unit ops and things like this. Feed is pumped into final module in a cascade, while diafiltration buffer is added to the first stage with retentate and permeate from the stage, recycle in the opposite direction. So let's look at what's happening here. So here is a diafiltration buffer, and it's coming in here, right? So the diafiltration buffer is coming in here, and it goes through this module like that, okay? Now our feed is entering at the other end, okay? So the feed comes through here, and then it comes back here as retentate, right? And then it's coming through here, and it goes into the second module, comes out, and goes into the first module. Now in the current system, we actually add the diafiltration buffer into the feed bag. But here what we're doing is we are contacting it with the feed that is coming to the third stage. So think of it in terms of a driving force. This feed will have the lowest concentration, right, of what we're trying to remove, but it's in contact with pure diluent. So you've maximized your driving force, right, because that makes sense. But this then comes out here, and you get a diafiltered product, okay? And then from here, the permeate here then becomes the diluent here, becomes the diluent here. But this already has some of this stuff we're trying to remove, but it doesn't matter because actually our feed is becoming more and more concentrated as we go this way because it's in a countercurrent action, do you see? So from a chemical engineering perspective, that's how we always design systems. And so it's rather strange that it took so long to come up with this idea in the biotech industry. But that actually is a very efficient method. And it can be shown that if you run a system like this, not only do you have continuous diafiltration, there's no recycling into a feed tank, but you also minimize your buffer usage significantly. So this is a three-stage countercurrent diafiltration. Feed enters the last module on the right, while the diafiltration buffer enters the first module. This is something that, again, has just been, it was proposed, I think, maybe like 3 or 4 years ago, very recently, and it's now at the pilot scale testing, but then you've got to get all this validation. But I'm sure that this will become commercialized fairly soon, because it's a very nice way to do this, okay? We can calculate things, so we can say the degree of impurity removal is given by here R, A, exponential beta, and the numbers are here. But the main point is this, that there's lower buffer usage, less fouling in dialysis mode. So it's actually a very nice system, okay? And it could be a single use as well. Use it and throw it away, okay? All right, one last thing. People have also come up these days with 3D printing, right? So there are also 3D printed modules now. This is another example of something that's been done, where basically this is a 3D printed diafiltration module for continuous addition and removal of the filtrate. So here's the feed coming through here. You're continuously adding your diafiltration buffer from this side and removing permeate from this side, okay? So that's another idea for doing this. This is not quite as efficient as that other one I showed you because it's not countercurrent flow. But it's nice because it's very cheap. It's 3D printed. It's very compact. It's all in one. That's another one that companies are working on and trying to sell, okay? So future directions here, I think I've covered this. It's a continuous biomanufacturing. There's a great deal of interest. For continuous operation, new module designs need to be validated, and of course, scale-up studies. These new module designs, when I say validated, validation in the biotechnology industry is a little bit different to other chemical industries. So now we have shown at the lab scale it works. We have actual commercial, if you like, or real modules, not just things we put together in the lab that you can test, like 3D printed modules. They work well. What's next? Well, if you were a biotech company, you would never say, something that's done at a, 1 liter, 2 liter scale. You'd want to see scale-up data. So what's next? You've got to build these now to say the next step would be maybe 10 liters, 100 liters, 1,000 liters, and so forth. Once you've got all that data, you then need to take that and go to FDA or whoever the regulatory authority is and get them to validate it and say that they accept this process. And then maybe people will buy it. So there's a long way to go, okay? But that's what's meant by scale-up studies required, okay? All right, any questions on that lecture? Because that is the first. So we had 2 lectures that were supposed to be talking about emerging therapeutics. And what I decided was I thought it's more interesting to talk about emerging technologies. That's emerging technologies. And the next one will be emerging therapeutics, okay? Yes?
I think it tangent to previous.
Yes, yes.
Can
You could.
If we have this, like if we, at the end of you say the filtrate,
Yes.
you said the flux will be lower because it might push back.
Yes.
So what if we use a single valve, like one side valve, it won't pass back. It has a diaphragm. It will
Yes.
only allow water passage to go once, not come back.
Right.
Then can we use that for like
You can.
efficient for the water?
It will be very efficient. The problem at the moment, I agree with you, it will be very efficient, and especially if you're using it for membrane bioreactor or something like this, right? It will be very efficient. The problem is that these are at the moment designed for biotech industry. So the cost is far too much. Okay? That's not to say you can't make it cheaper and it can't be done. But yes, it would be a very efficient system. But certainly as an experimental thing, it would be very nice to do. Yes, yes. And actually you can design your own, right? Because you just need a diaphragm pump and attach it to a membrane module, and that's all, right? So, yeah, I agree. Okay, should we take a, sorry, go ahead. Yes, yes. And that depends on the company, but you can't just put it in the garbage because all of these could potentially be hazardous. So yes, there are protocols. So you bring up a very good question. So now, is this actually environmentally better or not? And that is a difficult question to answer. I don't think anyone has really done the total life cycle analysis because there are many ways to look at it. So in the current process, right, you produce a huge amount of liquid waste that you have to dispose of. On the other hand, the modules you have are used for more cycles, right? So it has to be a life cycle analysis on all this. Compare that then to what happens when you have a system that you just use once and now you've got all this plastic waste, what do you do with it? But on the other hand, there is a significant saving in water, okay, in liquid, in buffers, in water, and all of that has to be made as well. Yes, there are protocols. Yes, this has to be considered. I'd say the biotech industry in general has been slow at looking at life cycle analysis as we do in water treatment or in manufactured biofuels, mainly because it's been driven by different things. It's been driven by health and safety. Okay. More recently, the cost has become a big thing. So now they're looking at these methods. But I would say that they are starting to look at life cycle analysis too, because many of our sponsors in our center are quite concerned about looking at the amount of buffer they waste, how much buffer they use, and looking at the overall life cycle of this. So I think it's coming. And what can I say? So I don't think there's a definite answer to this, but I suspect that overall, if there is an appropriate way to dispose of this plastic, it'll be much better than the current method. Yeah.
Where can we find the list of like what materials have been FDA approved? So there might be a list, right? So
There is a list. Actually, some of the references I used for this I can give you, so they would have them. But if you really want to, actually, you can go and search FDA guidelines for these things. So you can search for, say, microfiltration or membrane devices, and they'll tell you what has been approved. So regenerated cellulose, polyether sulfone, PVDF, so forth. There are lists, yes, for sure. Yes. Yes. Oh, the problem with the peristaltic pump is if you are trying to use continuous biomanufacturing, then your cells have to keep going back, okay? If it's a batch process, you don't care because you're harvesting and the cells are going to die and it's it. But if you want to send them back, the peristaltic pump basically there's a plastic tube, right, in the peristaltic pump. So basically it's got some rollers like this, right? And these are rolling around the tube, and it squeezes the tube, and in that way pushes liquid through. So this, between this and this, it pushes that liquid through, then the bit there, then the bit there. That's fine, but you're going to squash cells, okay? Now, it can be done. I mean, there are blood pumps, okay? And blood pumps are often peristaltic pumps, and they're designed to be able to pump blood, but basically they're designed to pump red blood cells, and red blood cells are highly flexible. See, they go through our veins, they go through the lungs, so it's okay. They are flexible and they're used to this. But these Chinese hamster ovary cells are not designed to be going through these. And so if you put a lot of shear stress on them, you can stress them, and then they don't grow well, they don't produce well. That's the disadvantage. So for continuous biomanufacturing, that's a problem. Okay?